in the processing steps now that we've gone through, you have a BAM file that has reads mapped to the reference genome. You have removed duplicates, and now we're going to talk about the next step. Life would be grand if we just had to deal with SNPs, but it turns out the genome is more complex than that, and there are other types of variation that are a lot more complex, and they cause problems downstream. So here's where we are in the flow. It's the local realignment, basically because of Indel issues. So what is the purpose here? When you have reads that are sequenced from a sample, and that a particular read overlaps a region where there's an indel in that sample, an insertion or a deletion, it turns out that it can wreak havoc with the alignments. So the aligners don't always do a perfect job aligning reads that overlap insertions or deletions, especially for larger events. And it turns out that these things can appear to be these artifactual SNPs, and you will misgenotype your samples. So better than explaining this to you in words, I'm just going to show you a screenshot in 1 second, an IGV screenshot that'll exactly describe this. So I hope that's not too blurry. And I'm going to try to use the pointer here to show you these things. So again, this is your IGV screenshot. There's lots of gray, lots of matches to the reference. But you see a whole bunch of columns here with these mismatches. Now we talked about before these SNPs. When you have these columns of mismatches, these look like beautiful SNPs. And over here you see some more SNPs here. Now it's kind of strange, though, that all the reads that we talked about directionality, all the reads in the forward strand have these SNPs at the end over here. But the ones on the reverse strand, they don't have the SNPs over there. They have SNPs over here, and not vice versa. So it's just very strange. This is a typical red flag. This is typical of an artifactual SNP. Something is going on here, and these aren't real. These aren't real SNPs. Something else is going on here. Notice here, I don't know if you can see it, there's TTTT, there's a little homopolymer on here. Homopolymers are notorious for causing true deletions and insertions in samples. Okay, so what is really going on here? Well, if you actually step back and take a look at things, this is what's really happening. There's a deletion here. And it happens to be a homozygous deletion. These reads don't show the deletion because they don't overlap the homopolymer directly. But there's a one base pair deletion. This is what a deletion looks like in IGV. Now, where are all the SNPs? They all went away. Once you account and incorporate the fact that these reads have a gap in them, well, all those other mismatches go away, right? And so it's very hard for an aligner, especially when the reads end at the right near the end of the deletion. So I'll say that again. When the reads end right near the deletion, it's very hard for them to decide, well, is there 2 SNPs or 1 deletion? And generally, it's more expensive to insert bases or remove bases from a read in their algorithm than to make a mismatch or 2 in the reads. So until you have the whole view of the whole picture, and you can see, as before, that there's a lot of weird variation going on, it's hard for you. We can't really fault the aligners there. But we can go back afterwards and reprocess the BAM and look for cases like this where the funky things are going on, they're inconsistent, and they're all clustered like this. And so this is the purpose of the Indel realignment step. It's to go back around these areas that are purported indels, either because some of the reads have already aligned with indels, or there's these clustered regions here, or there are sites of known variation, and go back and kind of realign using some Smith-Waterman hybrid to fix the BAM like this. Okay, so this is the purpose of this step. Principles. Okay, so I mentioned there's three types of things that can trigger the need for realignment. Now, not all of them are used at every single site. Most of the BAM file does not contain an indel, and just as you process it through this step, nothing happens. The read comes in, the read comes back out. But then you trigger at certain sites where there's fishy stuff going on. And what I mean by fishy, it's like I said, there's sites of known variation. You've seen in the cigar strings, the original cigar strings that we talked about, you see a gap, an insertion or deletion. Or, again, you have these, this entropy there is weird. There's a lot of clustered SNPs going on, like you saw in the previous screenshot. And so the main principle behind this is basically, given a pileup of reads, you look for potential haplotypes. There could be more than one, but we really, in any case here, only will realign around one alternate allele. So we'll say you can either be the reference, or it could be the indel. There can't be more than one possibility here. So if you're talking about regions that are STRs, these short tandem repeats, where you might have multiple deletions. You may have 4 base pair and a 2 base pair. That's not going to work over here. You need something else for that. You really need to deal with the haplotype caller, which Ryan will discuss tomorrow. But in cases that are kind of simpler, and that can be handled with a unified genotyper, you kind of look to divide the reads up into 2 different bins. One that should kind of are very clearly referenced and don't show any problems in them, and ones that are fishy. And you kind of divide up the fishy ones using an alignment algorithm, a local alignment, really Smith-Waterman, to kind of figure out whether there should be a gap or an insertion there. Okay? This is quality score aware. So the original alignments were not quality score aware. This is. This says, if all your bases are Q2s, which are really the lowest aluminum, Q2 basically means I have no idea, don't use them. Well, this won't use the Q2s. If they're Q10s, which means they're 90% confident, well, that'll mean a lot less than bases that are mismatching and are Q30s. You have a higher confidence in them. OK, so how do you actually run this? There's 2 steps. There's one sweep over the band file to find these regions that are interesting, fishy. And the other one is to actually do the processing, to create a new BAM file from the original one. So this creates a temporary file, an intervals file of interest. And this uses that file to then reprocess the BAM file. And we'll talk about each step individually. I think this is the first GATK tool here. So this is kind of the format of the further modules. You'll see the protocol and how to use them and kind of the flow. So flow, we start here with the original BAM file, and you run the realigner target creator. So the realigner it's the realignment process, and we're creating targets of interest for the realignment. Okay, and that's what we're doing here. And you're going to create this dot intervals file, just regions of interest. And this is the command line. We'd shown you these before. This is the typical command line where you have a reference sequence. Okay, you always need the reference sequence in all GTK commands. I don't care if you're running on BAMs, VCFs, doing nothing. You always need the reference. This is your original input BAM file. Sorry, someone is talking outside. This is your set of known variants. Okay, the list of known indels is not necessary if you don't have them. So if you're not dealing with human data, for example, then you don't need it. But it does speed up the process and it gets more accurate. We don't. As you process along, you might not realize that there's a hidden indel there that's artifactual. But if you have it in the list, well, it makes it much easier for us. And then it outputs these intervals of interest. This says here the BAM file is not necessary if you're processing only at known sites. We'll talk about that in a second. That's not recommended practice, but if you're really if you're working on very large-scale pipelines and you don't want to spend a lot of time processing, and you're not interested in necessarily ideal results, or you are using smarter SNP and indel callers downstream, which Ryan will discuss again with the haplotype caller, you probably only need realignment at known sites. You don't have to do the heavy-duty processing. You can do the quick and dirty processing. We'll discuss that in a second. Okay, so now you have your intervals file, and it's time for triggering realignment. And this step is a lot more computationally intensive. It's a little bit slower. This should be fast. This is a little bit slower because you're doing some work here and then rewriting a whole BAM file. If your input BAM file is large, well, then it's going to take some time to process it and to write out to disk again. Okay, and you can add known sites. Same thing here. So this is your command for the next step. This is triggering realignment. So again, you have your reference FASTA file sequence, your original BAM file, the same list of known indels if you have them, and then the intervals we got from the previous step, and the output BAM file. There are other optional arguments that aren't covered here. If you want a detailed technical description of everything, you can go to the documentation, which is linked in every single one of these files, and it'll describe all of the optional and extra or advanced options. OK, so we talked about all the possibilities here. So there's really 3 modes this can run in. There's another -mode argument, which is by default you shouldn't change it. But again, I mentioned known indels, which is much faster. It'll handle 90–95% of the indels in any given sample, because 90–95% of the indels are present in dbSNP. Of course, this is talking about human data. So if you have that data, you'll get most of them. You'll miss a few of them, and if you can handle that, it's no problem. Your IGV screenshots might not always look so nice, but it shouldn't affect variant calling too much, with one exception, which I'll discuss on the next slide. The default recommended mode is this. So you run with known sites plus any indels already seen in the original alignments, in the cigar strings from BWA. If BWA saw an indel, because let's say the read fell right in the middle and it was very obvious that there was a gap there. Well, we want to use that information also. And this is with new, larger reads, newer sequencers, better aligners, this is really all you need. There is a much more heavyweight mode, which is using Smith-Waterman in the alignments. This is what you need if you're still running, for example, Mac, which is BWA's predecessor. If you have shorter reads, like 36 base pair reads, older data, if you really have no known indels and everything is going against you, if you can't tolerate false positives, you really want to do the full heavyweight processing here. But again, it's going to take a lot longer, so keep in mind that that is a consideration. OK. And finally, you have your realigned BAM file. So at this point, you shouldn't have any artifactual issues there. So let me give you a cool illustration of results. This is very hard to read, unfortunately, but it's a diagram for our paper we wrote a couple years back. So the way to look at it is, let's look at just the first column. Okay, these are the before screenshots. The same site. This is from 1,000 genomes data with an older aligner and smaller, shorter reads. This is with GA2's Illumina GA2 data, and this is HiSeq, newer HiSeq data on Illumina with BWA, a better aligner. And you notice they have the same patterns here, regardless of the data or the aligner type. You see that there's these SNPs here, SNP here, a SNP here, and sometimes there's an indel here, a deletion here. All right, so it looks like this sample is heterozygous for the deletion for the two SNPs, and then maybe homozygous for the SNP over here. That's the before scots. Regardless, it's the same sample, but completely different data and aligners. Now you run it through the realignment process, and things look very different. Those SNPs go away. There's just a homozygous deletion. So these are just artifacts. Right? Now this SNP didn't go away. This SNP, even with the alignments, perfectly well suited towards this data. So it turns out that the true genotype here is that our sample is a homozygous variant for this deletion. And again, there's a little homopolymer on. So why is this a very extra cool screenshot? Because every single one of these variants, the deletion and all three SNPs, are present in our DB-SNP, our catalog of known variants. So you asked me, well, what does that mean? How could it possibly be? You just told me these are artifacts. Well, that's the problem with DB-SNP. People submit to DB-SNP, and sometimes they submit errors. They haven't done their realignment. They didn't do the right workflow, and they've submitted known variants that are actually not really true variants to DB-SNP. Now, DB-SNP is working hard. When we showed them this screenshot, they realized what was going on. They're working hard to filter these out. It's a non-trivial task. But just keep in mind, though, that if you're trying to look for concordance to DB-SNP, for example, you shouldn't expect 100% concordance because not everything in there is real. And that's OK. And I think Ryan tomorrow will show you some cool features, some kind of another example of where this shows up when you're trying to use these known sites for finding errors, errorful sites inside your output. Again, we can hold on that thought till tomorrow. OK. So now we're supposed to tell you, describe to you, did the step X really work appropriately? It's harder to do that with realignment. The one thing I'll say is that any read that gets realigned, we talked about every BAM record. Well, there's this metadata you can output. So we put in the metadata the original Sargar string. So presumably the original Sargar string did not have an insertion or deletion in there. It was just 100M, for example, just completely matched to the reference with no gaps. Well, after realignment, obviously there will be an insertion or deletion there because we have realigned it around an indel. So we put the original Sargar string in the OC tag. So if you want to if you're handy with Unix and you just cat the file and grep for OC, you can figure out how many and which reads were realigned by this tool. But besides actually going for the whole downstream processing, calling SNPs with and without realignment, it's hard to tell you that the realignment worked. But generally, this is a very stable tool. It's been run in the BroadE pipeline for many years now without complaint. So the best I can tell you is that it is working. OK, so one thing I mentioned before is that the best, the better SNP and indel mutation callers now are really running their own local assembly on the fly. So again, the unified genotyper, which Guillermo will talk about, is not. But the haplotype caller, our new and improved caller, which Ryan will discuss tomorrow, is. And so I would say for the haplotype caller, no, you don't really need to do this step. If you're just going to run the haplotype caller and all you care about is finding SNPs, this step in and of itself is not critical. However, the next part of this talk is about these base quality scores, which Ryan will talk about. We talked about the confidences per base coming out of the aligners. Well, those are not accurate. and you have to make them accurate. But if you, Ryan will, I'm making a note now, you will describe why in your presentation. If you don't do this step, if you haven't done realignment, at least at 99%, 5% of your sites, at these known sites, you will harm those results. You will get worse results. Okay, so if Ryan has not covered this, raise your hand and say, Ryan, tell us why do you need to do realignment? Okay, and again, as I mentioned, if you're running with the unified genotyper, for example, you definitely need to do realignment.
Because you only talk about BWA if you use other aligners.
You definitely, so the question was, we talked about BWA, what about other aligners? Yes, you need realignment. Other aligners, I actually presented this once as a poster, and authors of aligners came over to me and said, no, my aligner's better. I said, I bet you I can find you artifactual SNPs, give me a BAM file you've aligned, and I had no problem finding tons and tons of examples, and they all said, fine, we agree. Okay, maybe not all of them, but many of them. Okay, so this is where we were, and I think we have a break now, and then we will discuss the next step in the process.
So one question.
Oh, sure, questions, yes.
What's the maximum size of the Indel?
What's the maximum size of the Indel? So theoretically, there is no maximum size of the Indel. The Smith-Waterman can work on any size. You're capped by the size of the indels in your known catalog of variants. So in dbSNP, there aren't that many large indels. So that's one cap. The other thing is you talk about alignments already seen in the original, indels already seen in the original alignments from BWA. Well, BWA itself has kind of a cap on it, how much it can align. With a 100 base pair read, if you have a 50 base pair deletion, it's going to be very hard for BWA to align that indel. It's going to be very, very hard. So you typically don't see anything more than 20 base pair events inside the original alignments. The haplotype caller can actually recover a lot of this data and can call events that are larger, up to a couple hundred bases. But as far as realignment, yeah, basically capped by the indels you have in hand. So Smith-Waterman itself is going to fail at anything greater probably than 10 bases. So we're talking about the range of like 10 to 20 bases. Okay? Any other questions?
For the target interval creation step, which one option is better? For example, if you have multiple samples.
If you have many samples?
Many samples. It's better to create this interval sample by sample or create join with all samples.
Okay, that's a good question. So one thing it was on the slides, but I didn't mention, is that the inputs for the realigner target creator step, that first step, and the Indel realigner have to be identical. If however you've decided to group up your data, should be the same in both steps. So if you want to do it sample by sample, should be the same for both steps. Okay, that's step one. The other thing to point out, though, is we talked about these project designs. You might have lots of low coverage samples or one large sample. What you basically need here for the Indel realignment is enough data to kind of realize that something's going on. So if you have one sample and it only has 4x coverage, 4 reads per any base, you're not going to be able to do any realignment, really. It's just going to be too difficult because you don't have enough evidence of any events, especially if you have a heterozygous site where there's potentially only 2 reads. So what we generally advise people to do is if you have deep sequencing, so you have a few samples that have a lot of coverage, you'd run Indel realignment separately for each sample. If you have low pass sequencing, low coverage, then you'd group them up into little batches and run them through that way. Okay? The Indel realigner has an option to, if you have n files going out, it can keep the same n files coming out, or you can just merge them all into one big BAM file at the end. It doesn't matter. But you want to typically have like 30 to 50 reads per position with the data going in, and that'll be enough evidence for triggering it. Good question. Yep.
If you're providing a set of known SNPs, what about novel SNPs, those which are not in that list?
Right. So you're really providing a set of known indels. All this aligner is around indels. There is that heavyweight mode of Smith-Waterman. If you can't tolerate false positives, that will use known SNPs and kind of find regions of dbSNP where there's a lot of clustered SNPs because it thinks that can't really be, and it'll test that hypothesis. But generally, it's the known indels that you're looking at. Yep.
Do you see less of such a problem when you use, say, BWA SW, like the
Yeah, sure. BWA definitely has these issues still. Again, mostly because what you see, that original screenshot where every read was messed up, that's typically not what you see now. What you see is that any reads that overlap the indel, where the indel falls in the middle of the read, yeah, that's no problem. But when the indel falls on the end of the reads, you still have those issues. And so you need to kind of get a nice consensus there and make everything match up.
I have tried to use the BroadE instead of the but I think both of them are written by the same Lee.
No, Hung Lee didn't write BroadE.
Oh, did he?
Yeah.
Okay. Well, anyway, so I have never had any luck with the BroadE created band files.
Yeah.
Do you have any comments on that, or is there the tools can also fix the band file created by?
So BroadE, is that RNA-seq?
We could do both.
Yeah, so I will say this. It's a good point, and we should put this in the slides and make a note to say this. So this tool will not work on RNA-seq data. It doesn't handle large gaps, right? Because it's looking for indels. And if you're dealing with RNA-seq data, you already have these large gaps. It gets confused which are indels and which are indels that are supposed to be there, which ones are true variation. So this tool, as written now, can't handle RNA-seq data. I don't know. I can't speak for the rest of BroadE and for DNA sequencing data. But I will say, if your project is RNA-seq, don't use this tool. And we're not, it's not a focus of us to make it work for that because it's starting to get phased out given what Ryan will discuss probably tomorrow. Yep?
Is there any reason to go back to unmapped reads and try to realign them using this or it's possible to do that?
So the question was, is there any reason to go back to the unmapped reads? For this step, so if I understand correctly, you're saying go back to the reads that are unmapped, that never got mapped, and then given this, now you know there's true variation, remap them. Yes, some people do that. Not for this step. They go for when they're trying to find mutations. Generally, for shorter indels, most of the reads will get mapped, and I think Ryan will show you screenshots. What usually happens is that BWA will map the read up to the indel and then like soft clip off those bases so they're hidden. So they don't actually get unmapped. They just get like partially mapped and then cut. And Ryan's tool will uncut them and figure out what's really going on there. So you don't really need that. For larger events, one could do that, but it turns out to be not as trivial as you think. You really need to know the exact alleles in advance to be able to do this, and usually the breakpoints aren't quite as crisp or clear. So there is an aligner that does this. It's Andrew Kirby's. I forget what it's called, but he does that. He goes back to the unmapped reads and kind of does this realignment. It's very expensive to do that. So if you really can't miss your variant, you have a single target variant, yes, you might want to use his tool, but it's not something we necessarily need to do.
Is there an upper limit on coverage that you can handle with the realignment?
How much memory does your machine have? So the question was, is there an upper limit of coverage you can handle? Theoretically, if you can provide unlimited memory and compute, then no, there is no limit. But practically, if you're talking about a couple of gigs, we say the target is really 30 to 60x. It can handle a lot more than that. I think for the 1,000 Genomes project, we used to realign by cohort, which is 100 samples. Is that right, Ryan? 200 samples at 4x. We're talking about 800 reads there potentially.
I'm thinking about bacterial genomes where you might have 1,000x coverage or something, some ridiculous amount of coverage.
So 1,000x is actually not ridiculous. That's not so bad. Ami said he's dealing with 26,000 samples in an exome, so that's 26,000 times 100. So turns out, we're at different scales at this point. You just need to give it more memory and it should work. And there's actually a cap to how many reads you want it to use. All these are these other optional arguments you can deal with. Do we have time, by the way?
Yeah.
Okay. Question?
After realigning, do you remark?
I totally missed that question. So after realignment, do you? I missed that last part. Do you?
Mark duplicates again.
Oh, mark duplicates again. So I would say it's a great question. Again, because Mauricio mentioned that the cigar strings need to be identical for you to be able to mark them as duplicates. And based on the quirks or whims of BWA, you actually might have the same exact read that got aligned differently. So the answer is, in the official BroadE processing pipeline, they do mark duplicates again. So they do it twice. In our processing pipeline, the marginal difference in the calls at that point, it's so small, it's probably not worth the processing time. But the BroadE has a system set up in Picard where everything is just fast and it's automated, and they do it again. But I would say, for normal people, no, we don't really need to mark again. But it's a great question.
For the pair N, there's the duplicate rate is certainly lower than single rate. Single rate can be a lot higher duplicate. So for GATK purposes, does the program only look for the parent and duplicate reads, or any single reads that also do duplicates?
Are you talking about the marked duplicates, or are you talking about the Indel realignment?
No, the marked duplicates.
So marked duplicates here, so they do take into account the paired end reads. Is Mauricio still here? Yeah, you can confirm that. Can you confirm that? So marked duplicates is pair aware, right?
Yeah.
So the answer is yes. And you should know the Indel realigner, when it scores events, it'll ignore the duplicates, but when it actually realigns, it will realign all the duplicates. So if you do want to do this analysis where you want to keep the duplicates, you could do that. They are realigned.
Well, I guess what I was asking is, in the parent data, do you only ignore the duplicates that are parent and in both, or you actually only, or anything that's...
Any duplicate is ignored,
Any
right?
duplicate.
Yeah, it doesn't matter if it's paired end or single-ended. All right, so this module is done. I think we have a break now.